Okay, we're back, and to all of you hapless and helpless friends, we're going to talk about one type of molecule today, and one basically major contributor to that type of molecule. Okay, so basically I have a picture drawn on the board here, and some people all day when I was drawing it in front of my students were saying, okay, it looks like a pizza eater, okay, it looks like an upside down Pac-Man. I had all these sorts of jokes going on. Alright, really what we're going to talk about is one type of protein. Alright, so I tell my students in class, and you may remember this, enzymes are proteins. Proteins, a lot of times, are enzymes. Alright, so this class of protein we will talk about today is basically one of the largest groups of proteins in any living thing. Okay, we are talking about enzymes. This will be enzymes in general. So let's talk about this. Okay, reactions happen in the human body all the time. Thousands of times per second. Reactions are happening in the body. Got to get fuel into my cells. My cells have to function and do work. All right? I have to break down food to get it into smaller pieces so it can get to parts of my body. All of this cannot be done in many cases in the body without the help of these enzymes. All right? So I'm going to show you just a little demo right here. I've drawn a picture. Okay? This picture is of a disaccharide. This is a sugar. This is table sugar, okay? It's a hexagon, hexose sugar, and a pentose, a pentagon sugar. This is a disaccharide, means it is made of two simple sugars, like we talked in class. Okay, so this is a carbohydrate, all right? This double sugar cannot be used in our body, but both of these simple sugars can. And so what has to happen to this sugar for us to be able to use this sugar is it has to be broken into its two subunits. So what the enzyme does basically is comes along and helps me break this bond between the two subunits. Okay? And then I will have two simple sugars. All right? So what happens is, in that case, my scissors acted as the enzyme. They actually help to break the bond between these two sugars. And now I can use this glucose sugar, and I can use this fructose sugar, okay, to give my body fuel, to give my cells energy. All right? So, let's talk about this. Here I have my enzyme. I will label it as such. Okay, this is the enzyme. Obviously, this is also the same enzyme, and this too. Okay, this is what we call my substrate. Okay, my substrate, obviously, and this is important, has the same shape to fit in what we call right here the active site. The active site. Enzymes are proteins. We talked about proteins. We said proteins have a specific shape and it gives them a specific function. Well, the specific active site is called specific because it fits a specific substrate. In this case, the substrate looks like pizza. I made it up, okay? In the enzyme sucrase, it would break down that sucrose molecule. So it would kind of act like the scissors and break that into two. All right? And it would have an active site that is the same shape as that sucrose molecule. Okay? So in this case, my enzyme, its active site will interact with this substrate that has the same shape. Now, I drew this. Can you see the little teeth the little teeth going in. A lot of times when the active site receives a substrate, it changes shape to hold that enzyme a little tighter. Okay? It hugs it nice and gently and says, I'm not going to let you go until you break into the products that I am trying to break you into. Okay? So it hugs it tightly. All right? Once it does that, it kind of puts pressure on the bonds that hold the two parts together. And so in the end, at the end, the active site releases those two products, and I get product A and product B. And they can go off and be used in the body for what they need to be used for. The enzyme does not get used up. Okay? It can actually go back to the beginning and interact with another substrate and break it down into two more products. Okay? So when we say, what is the function of an enzyme? All right? Its main function is to speed up reactions that wouldn't normally happen or would happen too slowly on their own in the body. Okay? We can break the, maybe this can be broken down into two on its own in the body, but it might not happen fast enough to help me out, to get me the energy that I need. And so we have a helper to break them down much more quickly so that I can keep functioning. All right? Now, when we talk about enzymes like this, let's talk about one type of inhibition. Okay? So let's say the product, I get a whole bunch of product forming. All right? Well, with this whole bunch of product forming, sometimes the product can actually come back and interact with what we call an inhibition site or an allosteric site. All right? And with this, a lot of times, when it inhibits this enzyme, it shuts down the active site. Let's say that it gives it these gnarly claws and closes the active site. So if we have too much product, the body says, whoop, 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 we have enough product. We don't really need any more. But we still have substrate to be broken down. The body says, hey, you know what? One way we could stop this, let's just go and inhibit the enzyme. And so the product, let's say product A, there's a whole bunch of it floating around. And if there's a whole bunch of it, that tells the body there's too much. And it starts to be enough around to inhibit or interact with the enzyme to close that active site down. And it won't open back up until we have very little product left, and it stops inhibiting, it lets go, stops inhibiting, and then the enzyme starts functioning again and building more product. Okay, so they can be inhibited. A lot of times they're inhibited by the product that they make, so that when there's enough of it going around, it can go back, inhibit, basically, and stop the enzyme from making more until more is needed. All right? So what do enzymes do? They speed up reactions. How do they function? They have an active site. The active site is specific to one type of substrate. Okay? If the substrate doesn't fit in the active site in that spot, it won't work. It's specific. Okay? It helps to, a lot of times, break down a substrate into product. Also, don't forget this, at the end of this video right now, okay, we'll say this one last thing. It could take two product, okay, or two things here, and we could switch it and say this enzyme synthesizes things. It puts things together. Okay? So enzymes, we also have enzymes in our body that take two things and put them together to make something, to build things. So let's say one enzyme breaks down a protein that I eat into amino acids, and another enzyme takes those amino acids that I now have in my body, and it builds new protein with them, like in the muscle cells. That's an example. Okay, so that's what enzymes do.